Right. Good afternoon, everyone. So now that everyone has had some lunch, I'm sure I'm going to be noticing a lot of yawning and closing off. I'm going to try to stay up and try to go through these slides with all of you. And don't worry about something that you don't understand on the slides. I'll try to just hit the key points. There is no quiz after this presentation. If you have questions, feel free to ask points of clarification. I wanted to just provide measured hope, and that's the reason why that's the title of my talk here. You've already heard from several colleagues earlier this morning about where we've come from, where we're going. We're excited about so many new therapies, immunotherapies, CAR T-cell therapies. We're trying to still learn how best to utilize them for different settings in myeloma. But I'm going to put some of the framework around why we are very optimistic about the possibility of curing myeloma in the next decade or so. All right. Let's see. It is on. Here we go. Not this. No. All right, here we go. All right, so we'll start with this particular slide. So this is a slide that is very commonly utilized when we talk about what has been utilized to explain what the approach to frontline myeloma treatment is. We start patients on initial therapy, bless you. Typically, we used to use either 2 or 3 drugs in the past. I'll talk to you about what our current approach is in a bit, but then after starting initial therapy, we deem patients either eligible for a stem cell transplant or not. Patients get to a certain depth of response after initial therapy and a transplant, and then they move on to maintenance treatment, and they continue maintenance treatment until the myeloma comes back at some point, and then we think about other options at that time of disease coming back or disease relapse. But with this approach, patients who have the more vanilla flavor, standard risk disease as we call it, the average survival, overall survival for patients is about 13 years, just with that kind of an approach. That survival back in the late 90s, early 2000s, before the novel therapies started to come into the field, was about 2 to 3 years at an average. So we've come a long way, but we have a farther distance to go. For patients who have more aggressive or biologically what we call high-risk disease, where even after getting these treatments, there's a high likelihood that the disease will come back, for them, the survival is about 7 odd years, based on just getting 3 drugs with or without transplant, getting a single agent as maintenance, and then getting other options. And this information comes from different data sets, but for someone who would have been diagnosed with aggressive or high-risk kind of myeloma 20 years ago, the survivorship was even poorer. It was probably a year or less than that. So we've come a long way for all myeloma patients, but I think for high-risk patients specifically, we still need to have newer therapies that we can incorporate into our schema of treatment. So I'm going to just talk about certain general principles, what we're thinking about. I think the best chance of curing myeloma is when patients are diagnosed for the first time. That's when we can make the most impact, and we are working towards that goal. We are working towards figuring out where exactly in that spectrum of patients starting with precursor condition, what we call smoldering myeloma, to what we call active myeloma requiring treatment, we will end up drawing a line somewhere saying, if you have X, Y, and Z, we're going to treat you like you have active myeloma. And try to cure you or control the disease or functionally cure you. And there will be other precursor conditions where we would be able to say, all right, the likelihood that even if you have an M protein, even if you have some plasma cells in the bone marrow, you're not going to get into trouble with anything in the next 2, 3, 4 decades of life. You won't have any plasma cell problems. I think we are going to get there. We have much better tools in terms of looking at the cancer cell biology, studying the immune systems, studying what's happening in the bone marrow microenvironment. And we're trying to put all of that information together in a dynamic fashion, and that we're going to get there. The other important thing that we're going to accomplish is using better tools to assess depth of response. When we treat patients, we want all the cancer cells to go away. Right now, the assay that we utilize to try to educate on that is called MRD, or measurable residual disease. It's done on the bone marrow. It's either by flow cytometry or through next generation sequencing, as it's called. And these are just ways in which we try to identify the bad guys when patients are diagnosed, and then see if they're all gone. What we're learning is that the deeper responses can be observed in most of the patients in 1 in a million cells. If you count 1 million cells, you'll be able to detect the bad guys. And so there's a certain threshold that we want MRD to be achieved, and then we want that MRD to be sustained over time. And that sustained MRD negativity is what will eventually help us get to the cure. So it's not just kicking the bad guys down as low as they can go, but making sure they don't come down. So simple concept, but that's the only thing that this assay does. We are also going to eventually figure out whether we can do these MRD assays in the peripheral blood, and maybe we don't need a bone marrow biopsy for patients. So we're working towards that goal. This picture that I showed you, the previous picture, where we have induction or initial treatment, then we have some patients getting consolidative treatment with a transplant and then going to maintenance. Our goal now is to optimize each of those phases of treatment so that we can increase the number of patients who get to MRD negativity and sustain it. And that sustained MRD negativity threshold that we're thinking about, for standard risk patients, at least 2 years. For high risk patients, at least 3 years. And these are just, this is my view of how we're going to get there, but I think, this is, my team is following, these things as well. So we want this as the beginning or starting point to what we're trying to accomplish. And then hopefully we will continue to build on that. And then the last point is extremely important. So we want to increase the proportion of patients who get to cure without compromising on safety and quality of life. And this is where we have to learn how best to utilize treatments in a safe fashion. We have very effective technologies and treatments coming, many that have been discussed earlier today. We have to figure out what would be the best way to utilize them. So working on doses, how frequently to give treatments, all of that. So you don't have to memorize this part, but the way that we define high-risk disease, is written on these slides. This definition used to be different 5 years ago, 10 years ago, 15, 20 years ago. Dr. Davies is sitting in the back. She's played a very important role in helping get this consensus definition of high risk. And in 5 or 10 years from now, this definition may be very different, because we might be able to get certain treatment strategies going where we are able to overcome the poor prognosis conferred by one of these factors. But we'll get there. The reason why we need this definition is so that we can focus more on these patients, design clinical trials and develop therapies that will help us achieve that goal. And we have to do it in a concerted fashion just for these patients. Up to this point, the clinical trials and studies we've done has been just include everyone and see what happens. And now we really need to focus on these kind of things. So I think I already mentioned this part that right now the MRD assays that we have in myeloma are both flow cytometry and next-gen sequencing based, but the emerging assays in development are in the peripheral blood. But whenever we have these assays, we have to go through this exercise of proving that they're about the same or better than the existing assays in predicting outcomes, in prognosticating and predicting. So you go through this exercise of proving the point, and then when you get to a regulatory approval, when I say regulatory approval, these assays then need to have a very different benchmark of proving their scientific worth before the regulatory authorities like the FDA say, all right, we trust this assay. If we do this MRD, it's pretty close to the truth. That's what they're trying to educate on. So that whole process for the assays, the flow and the next generation sequencing assay, it took us as a field almost 15 years to convince the FDA that this is true. And that meeting happened earlier today, or earlier this year actually, sorry, where the FDA said, yeah, I think we trust what you guys are doing, I think this is fine. So we need another X number of years to prove the point for this peripheral blood assay. So they're coming, but I think once we have the right strategies for patients, we have the peripheral blood assays available, treatment, quality of life, all of those things will continue to get better for our patients. What can MRD do for us right now? Can we pick our initial treatment based on the likelihood of getting to undetectable levels of disease? I think we have enough information that yes, we can do that. Can we use it to stop treatment or do less treatment? No, we don't have that information yet. Can we use it to say, well, someone might need more treatment if they're not getting into MRD negativity? We can't do that either. Those are clinical trials and studies we're doing at the moment. So just something to, just to keep things back in your mind. Okay, I'm going to try to move through this slide and explain a little bit of it. Hang on, here we go. All right. So MRD negativity, the threshold that we aspire for our patients, I showed this slide, on the slide earlier, is 1 in a million, or even 1 in 10 million would be better than 1 in a million, but we want undetectable levels of myeloma cells. If we use 4-drug induction treatment as part of initial treatment for someone who would be going for a stem cell transplant, what is the MRD negativity rate that 4-drug regimen can get us to? It's about 20 to 27%. And these funny names that you're seeing, Griffin, Perseus, Ischia, all of these are the clinical trials that we've done to actually show that the 4-drug combinations are better than 3-drug combinations. So this data that I'm sharing with you, these numbers I'm sharing with you are just a summary of that information. So patients get initial induction treatment. 1 in 5 or 1 in 4 patients will get to MRD negativity at a very deep rate. If they get a transplant, we may be able to add between 15 to 20% more patients into MRD negativity at 10 to the minus 6. And then if we continue maintenance treatment with 2 drugs for them, for as long as it takes, about 2/3 of the patients will get to MRD negativity. And that translates into a survivorship of more than 15 or 18 years in majority of patients. So that 13 years of overall survival that I shared with you, We're already on the road to actually improve that even further. But the other sobering fact is a third of the patients are still not getting to MRD negativity even with these strategies. The number is still 65%, it's not 100%. So our work is still cut out for us. And what about sustaining that MRD negativity at the 12-month or higher mark? About half of our patients, about 47% patients get there, can sustain MRD negativity based on these clinical trials. It's not 100%. So my job is still not done. Dr. Davies' job is not done. We still have to work for you guys and do a better job. And for older patients who are getting a 4-drug treatment followed by 2-drug maintenance strategy, what is the MRD negativity for them? It's about 46% at 10 to the minus 6. And what about sustained MRD negativity? Now, the threshold for sustained MRD negativity that we have, in the public domain right now, it's actually 10 to the minus 5. So it's a little bit less sensitive. It's around less than half of the patients. So again, our job is not done. We still need to push the envelope for our patients. We've come a long way, but still have a long way to go. At MSK, we have modified our approach to treating initial patients with 4-drug regimens. Depending on whether they have standard risk or high-risk disease, we may use a different proteasome inhibitor called carfilzomib instead of bortezomib. But the bottom line is all our patients who are under the age of 70 would get a 4-drug regimen. We would prefer to give them a stem cell transplant, especially if they have high-risk disease. If they get to MRD negativity, a single agent as maintenance is fine. If they don't, then they need 2 drugs as maintenance. And that's our typical approach right now. We're becoming a little bit more cognizant about patients who are older than 70 and having broader discussions whether transplant is something that they would like to proceed with or collect their stem cells. But with the emerging data we have with the 4-drug combinations in patients who are older than 70, I'm, as of this year, becoming more comfortable deferring the stem cell transplant for those patients. So the reason for that is the 4-drug combinations followed by 2-drug. I can. I thought I was pretty loud and actually did not need a microphone at all, but I guess I do. In older patients, we have to start incorporating the frailty index. Not every patient who's older than 70 is the same. We carry different comorbidity baggage. We'll accumulate different health conditions as we get older, and people are going to be different. So frailty is very important. You cannot give 4 drugs to all patients. So based on the frailty score, we are going to be picking which patients can get 3 versus 4 drugs. Even with the 3-drug combination, the DRD combination that you see in this slide, if someone gets diagnosed at the age of 76 or 78 with myeloma, even if they just get 3 drugs started, their average survival is 8+ years. And then people like me are adding other options for them every year. So that survivorship will continue to get better. The folks who are fit or intermediate fit, their survivorship, even though they're older, is going to be 12 or 13+ years. So, it's, the field is changing, and that's the strategy that we're employing right now for our patients. The work ahead of us, myeloma is not one disease. So we have to figure out the timing of when different patients achieve MRD negativity, because that trajectory is different for different patients. And one question that was posed to me, I think, Terry, where are you? You had posed that question to me, that is it important when you get to MRD negativity, what we're learning is it really depends on the kind of myeloma you have. So there are certain types of myeloma where it might take you 2 years to get to MRD negativity, and you'll do fine. There are other myelomas that if you don't get to MRD negativity, you're going to be relapsing sooner. So, the achievement and sustainability of MRD negativity is going to be different for different patients. And we're just trying to figure that out. And we're going to have to incorporate newer therapies that are coming down the pipe, whether it's small molecules or CAR-Ts or bispecifics, in different scenarios, in different ways for standard versus high-risk patients. And that's how we'll be able to get to sustained MRD negativity and eventually stop treatment for patients. It's all right. And I know that there are folks here who have achieved MRD negativity and sustained it and have come off of treatment in this room as well. And that is the first step towards curing this disease. All right, so there will be a quiz on this slide because it's very simple. That was a joke. So, very complicated design, a very nice study designed by our French myeloma colleagues. And we are going to learn a lot from this. I would have done things a little differently in terms of the MRD threshold that they're looking at. But they're either escalating or de-escalating treatment based on MRD results. And we learned a lot of things from this study. We just learned a couple of interesting lessons. Even with this intensive kind of an approach, even with 6 cycles of 4 drugs as induction treatment, again, only about 2/3 of the patients are getting to MRD negativity, including high risk, including standard risk patients. The other important lesson, disease biology. So there are certain disease biologies that even after 6 months of treatment, it's not enough for those patients to get into MRD negativity. So when we think about consolidation strategies for different groups of patients, we have to think in a different way and design clinical trials in a different way. So we get there. And this will also be coming in the quiz, so please take a picture. All right, this is the future. If we look at just disease biology, some features of disease biology, some clinical characteristics, they're not useful in developing what I need to do with an individual patient. They can give me information about the whole population, but it won't be able to do that. And I think, Jenny, you actually showed this IRMA data set, but I want to share with you the breakdown of where those patients actually came from. So the IRMA risk model looked at incorporating biologic information, clinical information in patients who got 3 drug induction treatments. And this is data set from the MMRF COMPASS study, Moffitt, MSK, the German myeloma group, University of Arkansas. And these analyses were actually done at MSK. And that's where we'll be going. We are going to be looking at the same model with 4 drug combinations and building on this individualized risk model for multiple myeloma. But the patients that were included in the original model and paper that we published with our collaborators was different patient populations picked up from different studies at different periods of time. What we're going to do prospectively is have a uniformly treated patient population, gather all that data so that we can develop better modeling for our patients. So that's coming. I think Dr. Urvi Shah already talked about modifiable risk factors. So that's the precursor disease on newly diagnosed setting. I think I really enjoyed the lunch menu that we had today, by the way. It was very myeloma patient friendly. It was a nutrivention lunch. So on the way out, you can give your MRD specimens and microbiome specimens, but really, I think that's the new frontier. Can we modify these risk factors and change the outcome of patients? I think that's going to be a very important part of what we do. Incorporating immunotherapies earlier into that smoldering myeloma situation. So, my colleague Carleen Tan at MSK, she's actually looking at some bispecific combination studies and focusing on high-risk patients, using it for a fixed duration of time, and seeing if we can reverse what's happening to the immune system and bone marrow microenvironment of those patients. Then, for standard risk patients, my colleague Milan Cody, she's trying to optimize the induction treatment by incorporating a bispecific antibody. So increasing the rates of MRD negativity in that first 6 months, we know we only get 20 or 27% of the patients. Can we incorporate a bispecific and push that number over 50%? So this study is happening at MSK. For high-risk patients, we are trying to initiate a more comprehensive, Arkansas-like novel total therapy approach, where we are incorporating a CAR-T and a bispecific antibody into the schema of high-risk patients. I had shared with you that high-risk patients only get to about 7 odd years of median overall survival benefit. And we're trying to push that envelope here for our high-risk patients by giving them the best standard of care treatment and then adding on other things as consolidative strategies later. But the idea, you see a lot of MRD on that slide, and the idea is, can we push that cure fraction? Can we add more patients to sustain MRD negativity by incorporating these strategies? So hopefully we will triumph over high-risk myeloma, as the name of the trial suggests. And then CAR-Ts are being utilized in industry-sponsored trials to see if we can get CAR-Ts approved for frontline strategy. And CAR-T2-5 and CAR-T2-6 are two of those studies. They're open at MSK and at Sinai. There are certain subsets of myeloma that are very difficult to treat, and we simply do not have an answer for them, even with the best strategies we've got. Primary plasma cell leukemia is one of them. And so there's this clinical trial that we're developing in the US cooperative group mechanism, where we are going to be utilizing a response-based dynamic treatment model. It's going to incorporate some disease biology-specific treatments and some immunotherapies. It's an intensive treatment, but right now, this is like the most aggressive version of myeloma that anyone can get. There have been no clinical trials for this, and we are developing a clinical trial for that subset of patients. Historically, people who had plasma cell leukemia have always been excluded from clinical trials, and we are trying to initiate this trial specifically for those kind of patients. For standard risk patients, this is another, someone asked me this question actually, that, what's the role of transplant? We have these wonderful immune therapies, and, the cool thing about transplant is it's been part of that success story of pushing the envelope for patients. However, it may not be an option for everyone. Can we, for the longest time, high-dose melphalan was our best single agent. And now we have wonderful immune therapies that are giving better responses than transplant would have in the relapse setting. So why can't we combine them and use them as consolidation instead of a transplant? So this is a treatment strategy that we are pitching in this clinical trial, and Sarah Holstein. So I'm the chair of this, committee, the national committee, and multiple members from our committee are designing different trials that we're going to be running to answer these questions. So combining bispecifics instead of a transplant, can you get the same bang for the, or better bang for the buck in terms of MRD negativity, and then improve patient outcomes that way? And if the other aspect of this is, if we prove the point that bispecifics work as good or maybe better than transplant, then patients won't have to go to a transplant center. They can get the bispecific treatment, out in the community. That will elevate potentially the depth of response and outcomes for patients in Middle America, where for 300 miles there are no transplant centers. So, I mean, this is a forward-thinking, pragmatic study, and hopefully it will begin next year, and we'll be able to prove that point. And in the interest of time, since I'm already, a half a minute over, I'm going to skip over, I think, Dr. Milan Cody was going to highlight some of the work that he's already doing. I just want to give him a shout out. So he's trying to combine different targets for CAR T-cell therapies in this dual targeting clinical trial. Hopefully he'll report out some results next year. Sham, a shout out to you. And then he's also trying to sequence treatment. So giving telketamab after patients have received a BCMA CAR T to see if he can improve depth of response and outcomes for patients that way. And I'm going to skip over this one as well, but in fact I shouldn't. So what, the other way of trying to overcome the immune mechanisms of immune therapies not working, is boosting the T-cell health of patients. So this particular clinical trial at MSK being led by Dr. Hul CR, she's combining the CELMoD mezigdomide with a bispecific antibody and trying to boost the responses of patients, and then also studying the immune system. And if this strategy works for patients who are in the relapse setting, it will work even better in the frontline patients. So eventually, we'll try to see if we can move this forward into earlier lines. And I'm going to skip over this part and just share this slide one more time. So this is where I started my talk about how we can cure myeloma. I'm going to just leave this slide there and not repeat what I'm saying. But I think there's a lot of hope in the next decade. I truly appreciate everyone's patience in listening to me. And I'm not a funny guy, so thank you for laughing on my silly jokes.